So hello everyone, welcome back again. Today you can see here, today we will discuss about the regeneration process in case of various organisms. So regeneration, what is it? Regeneration is the sequence of morphogenetic event that restores the normal structure of an organ after its partial or total amputation. Amputation means it is the removal or the destruction by any means. Okay, it can be. So here we will discuss the regeneration in invertebrates. Regeneration in invertebrates. First of all, this is Hydra and Planaria, and last one is an annelid. It is also a invertebrate. Hydra, we are very much familiar with that Hydra, which is a representative of Coelenterata. A cross amputation in Hydra led to every part to regenerate to whole Hydra species. That means any part you dissect. In case of Hydra, it will give you that full adult 3 hydras if we make according to the sections. Planaria, we know the Planaria is a flatworm. Planaria contains neoblast cells which migrates towards the amputate or wherever we have cut the region and forms the losing part. Okay, and then annelids, the blastema. In this case, blastema forms. Blastema means it is a mass of cell that can ultimately form, that is going to form the limb or anything else, that is regenerated area of the amputate regions formed of ectoderm, mesoderm and neoblast cells which coordinate in regeneration of the losing part. Okay, so next we will see in case of Hydra. You can see here you can see this is a total Hydra. Okay, this is a total Hydra and it is cutted in this two portions. This is the foot portion and this is the head portion. You can see here from the head portion the foot is going to be regenerated here, and also let me take, and here you can see the foot is growing on, and ultimately here you can see the foot is in the 24th day and the 48th day it is totally grown as an adult. Okay, and here it is the foot portion and it is growing head. Okay, it is going to grow the head. Here you can see the tentacles are coming out. These are the tentacles. Okay, so next we will see in case of Planaria. Here again the amputate areas. Okay, these are the amputate areas. One amputate area, this is one amputate area, and there is another. That means ultimately it is going to give us 3 sections. We are considering 3 sections here. First it is, and second, and the third one in the 0 days. You can see here it is growing. In the second slide you can see here in this case the size is totally growing on the from the head portion. The tail is going to be grown. Here you can see the tail portion is grown, and this is the middle piece. From the middle portion the head and the back portion or the tail portion has been grown, and in that last portion where it is tail, it will grow the whole head portion, right? So you can see easily that these are the things which are promoting. Okay, these are the things which are promoting regeneration itself. In the mammals also the capacity of regeneration is very less. If we have a cut, the cut is going to be healed after some days, but if we cut a finger it will not grow like this, the organism. These are showing extensive the way of regeneration. Okay, in amphibia we will see. First here you can see the regeneration. It is a newt, that is amphibia only, which is having limb. Here you can see the growth cycle. Okay, the growth cycle here you can see this one. If we cut this one, the limb, okay, if we cut this limb, it will grow in 1 week like this. You can see here this is the blastema region, and in the 3 to 6 week it will grow like a normal leg, and ultimately the fingers or the toes, whatever we say, these are coming out from the matured limb. Okay, 6 to 9 weeks. Within 6 to 9 weeks, this is an extensive way of regeneration in amphibians, where they can develop limbs also. Okay, and in reptiles also, if you observe, only in case of where vertebrae is not present, in the tail region they will show the things. Okay, so regeneration of vertebrates. Okay, regeneration of vertebrates, we will see there are two types of regeneration. One is known as epimorphosis or epimorphic regeneration. So what is this? We will see here. This type of regeneration is involved in the reconstruction of the missing parts by local proliferation of the blastema or addition of parts to remaining piece. For example, regeneration of tail, limbs and lens in anurans, that is an amphibian, and urodels, amphibians also, and other vertebrates. Okay, so epimorphosis means the type of regeneration. It is involved in the reconstruction of the missing parts. This is the important thing. Missing part will grow, okay, and by the formation of the blastema. Okay, we should consider this term. That means we should remember this term, blastema. In the regeneration process, blastema is an important word where the tissue will grow into new additional parts. Okay, so next we will see morpholepsis or morpholeptic regeneration. Okay, this is the second type. In this type of regeneration, this is involving in the recognition of the remaining part of the body and of an animal. For example, in hydra that we have already seen in the diagram, hydra and planaria, they are showing the morpholepsis or morpholeptic regeneration. Okay, so very much clear about that, and we will go to the next, that is the epimorphosis or epimorphic regeneration. Tail in amphibians and reptile we will consider here. Amphibia, the tail lacks the vertebrae. As we know, vertebrae, what are the vertebrae? We have 33 segments, that means totally 28 vertebrae we have. But in case of amphibia, that vertebrae leg portion has an unsegmented cartilaginous tube which contains a regenerated spinal cord, okay, and which forms mainly the ependymal lining of the central canal. At the first, very few cells accumulated under the wound epithelium and the ependyma and the various connective tissues dermis. Okay, muscle septa, adipose tissue and osteocytes of the vertebrae are the sources of cell for the generating the new thing, that is the tail. Okay, so the non-nervous elements proliferated behind the apex of the tail, forming both the muscles and cartilage tube. Then the ependyma proliferates and gradually it extends dorsally. So we will see in the reptiles, for example in the lizard, the regenerated tail is a quite imperfect tail. It lacks the vertebrae and it is in place their place has an unsegmented cartilaginous tube. This tube contains a regenerated spinal cord. One thing we have to remember, that the spinal cord should be there. Okay, spinal cord, otherwise it should not be that means it is not going to be the animal cannot control the body part which is going to regenerate again. Okay, including the extension of the ependymal lining and the central canal of the spinal cord. Okay, right. So regeneration of the limb, we will see here 3 steps we will follow. First step is the phase of wound healing and the pre-blastema stage. Blood clotting and migration of the ependymal cells. Ependymal cells, we have to notice that ependymal cells are the very important cells in the process of regeneration. From the basal layer of the epidermis towards the center of the wound is covered with an epithelium which is thicker than the epidermis of the limb. Okay, we should consider this one. Number 2 phase of the blastema formation. I told you that blastema is an important word. The cell accumulation beneath the epithelium covering formed the blastema. Mesenchymal cells accumulated beneath the cap, mesenchyma, blastema. Cells differentiated into myoblast and the muscle cells, early cartilage cells and cartilage. During the dedifferentiated phase of hyaluronate, increase in the distal stump of the blastema. As the blastema forms, the hyaluronate, HA means hyaluronate, will be decreased. The population, the production of the HA, hyaluronate, and breakdown of collagen represents, collagen we know this is a fiber, type of fiber. Connective tissue comes under the connective tissue, represent the establishment of the migration of stumps tissue, okay, which is going to form the limb. The phase of the dedifferentiated and morphogenesis. Now morphogenesis, the blastema begins to restore the part of which the limb has deprived. Specifically, the if there is a forearm is removed, the blastema differentiated directly into the muscle, bone, cartilage and skin of the forearm only. Okay, if it is that means hind leg or hind arm, it will be going to form that hind one. Okay, So steps of regeneration of limbs, we will see here in one diagram: amputation, wound healing, dedifferentiation. Okay, amputation means the cut. Here you can see, here you can see amputation. Okay, then wound healing and dedifferentiation. Early bird, here you can see after amputation, wound healing and dedifferentiation. Okay, early bird will come out. This is the median bird and the late bird. You can see the structure. Okay, basically you can see the structure. This is the palate and early differentiation, early dedifferentiation, and the complete. Okay, after dedifferentiation, this is the dedifferentiation. You can see steps of regeneration. Here one GIF file, you can see here how it is growing on the limb. Okay, so clear pretty much steps of regeneration. Factors controlling regeneration: these are the nervous system, animal size, and pituitary glands. Vitamin A is and its derivatives. As we know, the provitamin of vitamin A is retinol, and beta carotene, sorry, retinol is the name, and the insulin also important for the controlling of regeneration process. So thank you. We will discuss these things extend, and one thing is that those who are not in the sector of biology, they will not get this one, and I have made this style slides for making some notes for the BSc students, and the class 11-12 students also can get the knowledge. Okay, so thank you everyone.